right, AP Bio, and welcome to our lecture on bacteria and the origins of life. So we're going to be covering chapters 24 and 25 in this one from your reading guide. So most of what we've been talking about so far when we talk about cells and we talk about the organelles, we've been talking about eukaryotic cells. Eukaryote, again, means true center or true kernel. That's referring to the nucleus. They all have a nucleus, they have membrane-bound organelles, and while it's only one of three major domains of life, there's 4 to 5 kingdoms. So protists, fungi, plants, animals. Most of the life that you see and interact with, that you can physically see, are eukaryotic cells. Prokaryotes, on the other hand, meaning before the center, they have no nucleus or no membrane-bound organelles, but they still make up a vast amount of the life and biodiversity on our planet. And so today we're going to dive a little bit deeper into what makes prokaryotes who they are. So you may be asking, if we evolved into eukaryotes because organelles give us the ability to do so much more, why would you ever stay a tiny little prokaryote? Well, it's actually kind of a big hurdle to make that jump because in order to have organelles, you need to be a lot bigger. And as you grow in size, it actually becomes more difficult to survive. So when we talked about diffusion, most cells, or all cells, rely on the ability of diffusion to move stuff around. And as you get bigger, so eukaryotes are much bigger than prokaryotes, your surface area increases much more slowly than your volume does. So here we have 4 cubes with an edge. Each edge is 2 centimeters, 4 centimeters, 6 centimeters, and 8 centimeters. And as you can see, the surface area will increase by a square, so 24, 96, 216, 384. But volume is increasing cubically. So it's 8, 64, 216. It used to be behind, now it's the same. 512, now the volume is a bigger value than your surface area. So that surface area to volume ratio keeps shrinking as you get bigger. And so if you think you need more stuff inside you to support a bigger cell, you need more energy, more nutrients. But the only way that stuff can get in is through the cell membrane. And so the more membrane you have, the more openings you have for stuff to get inside and spread out to where it needs to go. But if your surface area isn't growing as fast as your volume, your needs are increasing faster than your ability to bring it in. And so that's why it's harder for cells to get bigger. And so until they were able to compartmentalize with organelles, they really couldn't grow to the size of eukaryotic cells. And so that's why it's kind of a evolutionary hurdle to get past in order to grow to that size. So let's look at what's inside a bacteria or a prokaryote. So prokaryotes do not have a nucleus. It's before nucleus is formed. So they have what's called a nucleoid, where all their it's just a general region in the center where all their DNA is collected. And their DNA is going to be a circular shape instead of the linear DNA that eukaryotes have. So it's going to be a big loop. Surrounding that nucleoid is the cytoplasm, which is filled with ribosomes. None of those ribosomes are bound because there's no membrane system. So there's no ER. They're just free floating everywhere. Then you have a cytoplasmic membrane wrapping that. And then outside of that, you have a cell wall with peptidoglycan as the structure holding that in. And then outside of that, you have a capsule. Now, most bacteria are going to have these structures that you see called pili, these little hair structures, and that's going to allow for them to hold on to surfaces, to grab other bacteria, and exchange genetic information and interact with their population. Now, these bacteria can perform a lot of the functions of organelles, but it's going to be a lot slower because it's just diffusing through the cytoplasm instead of what organelles do, which is keep things compartmentalized. So you have all your acids in one place, all your enzymes in another place. Now they're spread out, so things take a little longer. So how do they do all that cool cell stuff? Well, the answer is typically, well, it happens in the cytoplasm. So instead of that nucleus, they have the nucleoid, that region where their big loop of DNA is all bunched up together. Instead of an endoplasmic reticulum and a Golgi apparatus moving things around, they just use diffusion in the cytoplasm and wait for things to spread out. Their ribosomes are all over the place to grab things. And then they'll also have tiny structures of DNA, accessory genes called plasmids that are smaller loops in their cytoplasm that can also be encoded for different proteins. So let's talk about the two domains. There's three domains of life: eukaryotes, bacteria, and archaea. And so two of our three domains of life are all prokaryotic cells. And they're going to be unicellular also. Now, bacteria can be heterotrophs. They eat other things. Autotrophs, they make their own energy. Or saprotrophs, they eat dead material. One of the biggest decomposers in the world is bacteria. As I mentioned, they have peptidoglycan. So when you see that word peptidoglycan, if we break that down to peptide and glycan, you can see peptides, meaning protein, and glycan, which is going to be implying sugar, like glycemic. And so we have both carbohydrate and protein together, making those cell walls. Archaea, on the other hand, also prokaryotes, also unicellular, also heterotrophic, autotrophic, and saprotrophic, but they typically live in extreme environments. So here you can see the prismatic pool, really famous hot spring in Yellowstone, where these different bands of colors come from different archaea or extremophile bacteria that are, some of them are anaerobic, some of them are aerobic, and them performing different functions makes different colors based on what they're eating and what they're producing. So archaea are going to be very distinct, and in fact, they're actually more related to eukaryotes than they are bacteria. So a big thing that we're going to see that's different as well is because of their difference in genetic structure, they don't have a nucleus, they don't have as much protection, they're going to reproduce differently than our cells. So our cells, you're going to copy the DNA, check to make sure everything's good, break down the nucleus, and then make a cell division. Prokaryotes don't have time for that, and they do everything simultaneously. So the DNA is getting copied while the cell is splitting, while it's breaking apart. Those chromosomes are getting reallocated. And so it's going to be all asexual division, this binary fission, where everything's happening at the same time. There's no steps. You just do it all at once. But that does raise the question, if bacteria are all producing asexually, how do bacteria create diverse exchanges of genetic information so that they have a diverse population that can evolve and survive disturbances? Because you need diversity to survive disturbances. Well, bacteria have evolved a few different strategies for this. So as I mentioned earlier, bacteria have plasmids, which are accessory or extra genes that are separate from their main genome in the nucleoid. And these are smaller loops that just maybe have a few genes encoded on them instead of all the genes. And a lot of times these plasmids give a bacteria extra abilities that they may not have originally had, or they come from outside sources, not their original binary fission material. So how do they get them? Well, the first one is conjugation, which is the most similar to sexual reproduction that bacteria can get because two bacteria interact and exchange genetic information. So they use their pili, and one bacteria will grab another one with their pilus, and they're going to bring them in close and make a connection between their membranes. Then the donor is going to make a copy of one of its plasmid and insert that into the other bacteria. Then they're going to disengage, and now both bacteria have those plasmids. So say that plasmid is something like antibiotic resistance. If only one bacteria has it, if antibiotics come in, only that one bacteria will survive. But if it's able to pass this gene along because it's favorable, now a larger percentage of the population is going to have these genes. So it's all about maximizing gene sharing to maximize genetic diversity in the population. So I love bad analogies, so I'm going to give you a bad conjugation analogy. Genes are the code for doing things. A new gene allows you to do something new in your cell. And so since we live in a horribly capitalistic society, it's sort of like how money lets us do things in our world. And so conjugation would be like one bacteria handing another bacteria $5 so it can do something it previously could not, like go to a movie. That would be its protein that it's now making, the ability to see a movie. Like I said, it's not great. So the next one that bacteria can do is called transformation. So when a bacteria dies, frequently that cell membrane will rupture and some of that DNA will spill out. And so either plasmids or the original chromosomal DNA may break, it'll start to break apart. And so there'll just be these fragments, but those fragments can contain entire genes. If a new bacteria comes along, it can uptake that naked DNA or that free-floating DNA, and then it can either turn that into a plasmid or recombine that with its own genome. And so you can see here, like the gene big B here is incorporated into the main genome. And now it has new genes. So it's a more diverse population. So back to our bad money analogy, someone has died on the street, and when they died, they dropped $5. You come along, ignore the body, but you pick up that $5. You step over the body to walk into a movie theater, and now you can go see a movie because you have the new gene or the new means of doing something. The last form is called transduction. So watch the video on viruses to learn more about bacteriophages. But what viruses do is they infect a cell, insert their DNA, that gets incorporated into the host DNA, and they kind of take over or hijack the cell and turn it into a bacteria-making factory. Those virus-making factory. Those new viruses now will have some of the DNA that was copied, and they'll move on to a new cell to infect it. Now, it's not only virus DNA that gets packaged into these viruses, though. Sometimes bacterial DNA will also get packaged into these viruses. So a virus infects bacteria A, that DNA gets integrated, breaks down the bacterial chromosome, the virus takes over the cell and makes all these new viruses, some of which have viral DNA, some of which have bacterial DNA, some might have both. Those new viruses will break free and they will go infect new cells. If one of those cells infecting a bacteria B, if a virus with bacterial A DNA infects bacteria B, that's going to get incorporated, and now that bacteria has new genes. So back to my bad analogy with money. You have $5, and your kid comes along, the virus comes along, and steals $5 from your purse. Then that kid, or again, the virus, gives that $5 to a friend, so now they can see a movie. So stealing your DNA or your money and passing it along to someone else so they can do what you would have been able to do with that $5 or with that gene. So hopefully that cements in your mind conjugation, transformation, and transduction. So we know that prokaryotes are simpler than eukaryotes, but how did they come about? How did life first start? So we know that the origins of life were really chaotic. There was not really a breathable atmosphere. It was filled with noxious gases like ammonia and methane and hydrogen gas. There was no oxygen because photosynthesis hadn't come along to make oxygen yet. And there was a lot of storms, so lightning strikes. There were thermal vents erupting. There wasn't much of an atmosphere, so there were a lot of meteor strikes and radiation coming in. It was a chaotic world. And so Stanley Miller and Frank Urey at the University of Chicago in the 50s made an experiment where what they did was they had a contained system where they heated up some water to simulate the ocean getting heated up and evaporating into water vapor into the sky. And then that water vapor would evaporate up into a container with a simulated early Earth atmosphere with methane and ammonia and hydrogen gas. And then they applied energy to it, simulating lightning strikes or thermal vents or anything else that would provide energy. some sort of combustion reaction, and they collected the condensed water as it cooled, and they ran it through a filter to get whatever came out. And what they found were that they collected amino acids. So from nothing but the basic ingredients of what was there on the early Earth and applying some energy, they were able to make out of nothing amino acids, which is really important because, as we know, amino acids are the building blocks for proteins. This experiment was then replicated years later with ice core samples in Antarctica, which had trapped compounds from early Earth atmosphere in it. And they were able to replicate the same experiment, and nucleotides formed, so the building blocks for DNA and RNA. So this was really exciting to say that, yes, it is possible that the building blocks of life could form. But how do we go from chemistry to biology? How do we go from unliving to living? So that's where we need to look at the original life molecule, and that's going to be our RNA world hypothesis, or the RNA world theory. And in the 1980s, two researchers, Sidney Altman and Thomas Cech, discovered that RNA, ribonucleic acid, can catalyze chemical reactions. So this is known as ribozymes, where those nucleic acids are able to carry out the same function as enzymatic proteins. And we also knew that early forms of life used RNA to store genetic information and also catalyze some reactions. There's a really great video on the RNA world theory that I'm going to post that I also suggest you watch from Stated Clearly. They do a really great job of breaking this down. But here's basically what happened. So we're starting with Miller and Urey's prebiotic soup, so that primordial soup filled with these macromolecules. And as energy is applied, they start to combine into polymers. So we have the spontaneous generation of amino acids and nucleotides. Then favorable chemical reactions make them form long chains of RNA or similar structures. And these RNA chains are capable of self-replication. So one strand of RNA acts as a template. It grabs other nucleotides to make a corresponding strand, and now there's two strands of RNA. When it's heated, they'll break apart, and now those two strands can make copies and make copies and make copies, sort of like how PCR works. And so over time, there's mutations, and so these RNA chains are changing slightly, and as they change, they're capable of doing different things. So those free unbound nucleotides of RNA are also capable of grabbing substrate and catalyzing reactions. So this is what's called a ribozyme, where RNA is working as an enzyme. And as more mutations happen, the capabilities of ribozymes are going to change. And eventually, researchers have found when they simulated this, within a few generations, ribozymes were formed that are capable of building nucleotides with surrounding materials. So if you're a better competitor, you can make what you need to make more of yourself. You're going to explode in population, and you're going to be the dominant feature. We can see forces of evolution and natural selection at work here. And so RNA would lead to DNA as you form double strand instead of single strand, which would lead to proteins to help out. If you think about the structure of a ribosome, which makes proteins, it's part protein and part RNA. So we can see that this progenitor of all life, this RNA, building proteins, making itself a ribosome to help out. Eventually, lipids could form, and as we know, lipids will form like micelles or membranes. They'll form little bubbles, and if RNA could get into a bubble, now it has a lipid shelter, and now we have what's basically a prokaryotic cell, a membrane surrounding some self-replicating genetic material. So it's really exciting. So this is how we form our first cell. And as this continues to evolve, eventually we reach LUCA, or our last universal common ancestor. Now this means this is the last in the stage of evolution of life. This is the last organism that all life today can trace its lineage back to. So LUCA, all life that exists today, the family tree goes back towards LUCA. Now there may have been organisms before LUCA, But LUCA is where it branched off into bacteria and what went towards eukaryotes. So we can't really be sure what LUCA looked like, but we know it was a heterotroph. It ate other things. And how do we know it wasn't photosynthetic? Well, not everything alive today can photosynthesize. So it's very unlikely that such a powerful ability like photosynthesis was lost. So if we think back to our cladograms and think back to parsimony, it's most likely that it couldn't photosynthesize. Because there's so much life in bacteria and archaea that are unicellular, it's very unlikely that it was multicellular. Since all life uses DNA, it probably by this point had moved from RNA to DNA, and so probably used DNA. And since photosynthesis hadn't existed yet to make our atmosphere filled with oxygen, it was probably anaerobic as well. And so here you can see a family tree with LUCA branching off to bacteria, and then branching off towards archaea, which are actually more closely related to eukaryotes. And probably there was a branching point where something stayed archaea, and then something gained the ability organelles to become eukaryotes. So eukaryotes, again, are protists, plants, fungi, and animals. And how did we make this leap? How did we move from prokaryotes to eukaryotes? We know that size is an issue. How did we overcome that? Well, through organelles. How did organelles form? Well, something called the endosymbiosis or the endosymbiont theory. So things like the nucleus and the endoplasmic reticulum, which are just folded membrane-bound sacs, basically, those probably happened from a cell doing endocytosis or eating something, and bits of membrane came inside, so it folded in, and those infolded membranes surrounded the nucleoid to make a nucleus, probably created these passageways of the endoplasmic reticulum. And then we would get other organelles, like mitochondria were actually their own bacteria. They were their own prokaryotes that were swallowed by a larger cell. And instead of breaking it down for its nutrients, this cell found that mitochondria are actually way better at making energy. So instead of breaking it down, it just left it there. The mitochondria liked it because it had protection. Eventually, after absorbing mitochondria, this cell would go on to engulf, or some copies of this cell would go on to engulf another prokaryote that became chloroplasts, and it was photosynthetic, which was another great superpower to suddenly have. And so this was the break of what would then evolve towards plants versus what would eventually evolve towards animals and fungi. So this was the breaking point. But it all comes from swallowing other organisms and not killing them, but leaving them inside. And what are some of the proof we have of this? Well, chloroplasts and mitochondria both have their own set of ribosomes. They both have their own DNA. They have double membranes, which means that they had a membrane, and then when they got engulfed, got another one. And you can tell because those membranes have different components. So there's the ancestral bacterial membrane on the inside, and then one that resembles more the cell that ate it. And this is really cool when we look at our own cells, because mitochondria actually reproduce themselves. They're not divided. And if you were to remove all the mitochondria from a human cell, it couldn't make new ones because mitochondria is its own organism inside all our cells. And your very first mitochondria came from your mom, because when an egg and a sperm fuse, the sperm is basically just DNA and the egg has all the organelles. So that means all your mitochondria came from your mom. You have the exact same mitochondria as your mother, who has the exact same mitochondria as her mother. And since mitochondria have their own DNA, this means you actually have more DNA in your body from your mom than from your dad. So you're a little bit more your mom than your dad. Pretty cool. So that's our lecture on bacteria and the origins of life. Some take-home concepts. Make sure you're familiar with the origin of life, that RNA world hypothesis. The AP test sometimes loves to talk about it. Same with that Miller and Urey experiment. Is a very famous experiment, so you should be familiar with it. You should know basic bacterial anatomy, the nucleoid, pili, that there's free-floating ribosomes, and that everything happens in the cytoplasm. You should be familiar with bacterial reproduction, so you should know how binary fission works, where it's all happening at the same time while the cell splits. And you should be familiar with the terms conjugation, transduction, and transformation, or how bacteria exchange genetic information. And finally, you should be very familiar with the endosymbiont theory and endosymbiosis. I don't know that there's ever been an AP test that didn't ask about endosymbiosis at least once, so you should definitely be familiar with that. All right, come back next time for our lecture on viruses.